Okay, guys, good morning. So today's class is actually most exciting for me because I'm going to discuss what my lab does. Last few classes were about materials, the issues with medical devices, fouling of medical devices, infection, clotting. Now, my lab is working on a lot of things to try to mitigate the issues with the current medical devices. Okay. Also, please remind me that in the afternoon session, 15 minutes or 20 minutes before the session ends, we should stop talking about this and talk about tomorrow's exam. Okay. Tomorrow morning is your 2-hour exam. Okay. Let's start. So, conquering infections and thrombosis through biomaterial innovation. Before I start, I want to tell you guys a little bit about my background. Why am I doing what I'm doing now? A little bit about my lab. So, I did my PhD under Dr. Gongzhao Mao in material science, surface chemistry. Lot of atomic force microscopy, understanding coatings and films. Now, Dr. Mao is now the school head at University of Edinburgh, and I'm very grateful to her because when I went to her, I had just finished my undergrad from DCE, very minimum lab experience, and she was so patient with me and she taught me so much. Now, after I finished PhD under Dr. Mao, I went to medical device company. I thought if you are a professor, you have to do very basic mechanistic research, but I liked a little bit more practical research. So I thought if I go to industry, that's where I can do all that. If I become a faculty member, I have to do mechanistic research. Now, I worked for a medical device company for 2 years on developing nitric oxide releasing coatings for catheters and other implants. But while working there, I met with Dr. Bartlett and Dr. Meyerhoff. Dr. Bartlett was a co-founder of the company that I was working for, and Dr. Meyerhoff was scientific advisor to the company. They both were professors at the University of Michigan. Dr. Bartlett is a retired surgeon now, also known as father of ECMO. Dr. Meyerhoff is a leader. He also retired last year, but leader in nitric oxide releasing materials, chemist by training. So Dr. Meyerhoff was a chemist, Dr. Bartlett was a surgeon, and I had an engineering background. So after 2 years at the medical device company, they hired me at University of Michigan in Department of Surgery. And I think that was I'm happy, that was a good decision that I made to go to work with them. Because I learned a lot from them also, and they are the reason what I'm doing right now. While working with Dr. Bartlett, Dr. Meyerhoff, I learned a lot of animal models. I learned a lot of chemistry. I learned a lot about the clinical issues that surgeons are having with the patients. So working with them was a great decision for me. Now, little bit about my lab. As you know, I'm at the University of Georgia. 2 years ago, they built this new building called STEM building. And this first picture is the building from outside. The second picture on top, that's my lab where the students sit. So that's the student office area, the cubicles. This is a conference room. So the building has a lot of such conference rooms because they want to really promote collaboration between the students. So there are a lot of rooms bigger than this one, smaller than this one, to encourage students to come and sit together, discuss projects, just to promote more collaborative discussions. And the bottom last picture is our kitchen outside our lab, where we interact sometimes or have lunch. And So really nice building, great atmosphere, very bright. So I'm lucky that we were able to get lab in this building. This is my lab. So we have almost 7,000 square feet lab space. There are a lot of support rooms like that one, the right picture on the top and bottom, both right pictures they are. So we have like a separate room for cell culture, for virus work, for bacteria work, for blood. One of these rooms we want to convert it to a surgery room. We just need to do lot of documentation for that, and then we will be able to do that. We have almost 25 fume hoods to work. So great place to work, and a great team. I'm very thankful for the students I have who are doing great work. So lab capabilities, we will talk more about the research, but mostly my lab has people with chemistry background, microbiology background, pharmacy background, and of course lot of engineering background, chemical engineering, polymers, biomedical engineering, veterinarian, because we do a lot of animal studies. The goal is that we can synthesize new materials, design new surfaces, then we can do a lot of characterization. It can be AFM, SEM, FTIR, NMR, XPS, to make sure whatever chemistry you did or whatever new materials we are synthesizing, molecules, they are pure and they are what we think they are. Then we do nitric oxide releasing testing. Some of the tests we talk about, sterilization, leaching studies, all those we do. Then if those results look good, our surfaces look great, materials look great, we do a lot of in vitro studies, which are like protein adhesion studies, bacteria studies, blood studies, virus, if we are working with virus, cytotoxicity. So we work in a lot of those things. And if those results look good and the technology is very promising, we have animal models. Either we do it, we have done rabbit and mouse models in university. We can also do pig and sheep in the university, but I have collaborators who can do pig and sheep studies for us also. So translational research, by now you already know what is translational research. So when I was starting my lab 10 years ago at the university, I wanted to try to make it as translational as possible. Translational research is from benchtop to bedside. You're designing a surface, you are doing the testing, you're going to the animal study, and then you're trying to commercialize the material. And that's what we are doing. So we have development phase where we are making coatings, surfaces, catheters, wound dressings. We are doing all the characterizations, imaging, all that is looking great. Then we are doing hemocompatibility, antimicrobial, ELISA assays, toxicity studies. Those are looking great. Then we move to catheter studies, ECC models that I'm going to show today. If those are looking good and the work we feel is very novel, we file for patents, patent disclosures to the university, and we test those materials in animal studies. And whatever promising technologies are coming from the lab, I have a medical device startup company called Nitrix. We try to move those technologies and license the patents from the university in the company so we can try to commercialize the product. We showed this picture before also. I got this image from Dr. James Breyer. Basically, we have so many different implants going on, right? So what I feel is, if we can make a truly antimicrobial surface or hemocompatible surface, there are unlimited applications for that material. Because a catheter coating that's really antimicrobial can also be used for other polymeric surfaces that are getting implanted. So one success for one application can actually be extended to so many different applications, can open a lot of avenues for other implants. My lab works on a lot of different areas. Last night I was trying to, I'm going to show you some of the work we are doing, but I couldn't collect everything. But I'll be more than happy to discuss with you. Email me, I can send you papers if anything is exciting. On the website, you can see my website. If any paper looks good and you're interested, I can send you those papers. Mostly how it started was, I told you Dr. Bartlett is father of ECMO, or extracorporeal life support. And when a baby is on this ECMO, the baby has to be heavily heparinized. You can use ECLS even for adults also, but from Dr. Bartlett's perspective, he was doing it for babies, right? And you have to heparinize the patient. Heparin is making blood very thin. And even after you heparinize the patient, this is what's happening. All this black, still clotting is happening. A lot of clotting is happening. And people can be allergic to heparin also. So Dr. Bartlett, Dr. Meyerhoff, when I was working with them, the vision was, can we make a surface that we don't have to give heparin? Can we coat all the surfaces in our ECMO circuitry that we don't have to heparinize the patient? Or reduce heparin dose a lot so that we can prevent clotting of those medical devices. So that's how it all started for me. This also you already know now, and maybe this is a good revision for you guys for tomorrow's test, that if you have any medical implant, proteins adsorb on the surface very rapidly, platelets get activated, adhere to those proteins, they call more platelets, they form cross-link, fibrinogen gets converted to fibrin, some red blood cells also get trapped in it, and now it leads to a clot formation, right? And all this is happening within seconds to hours, very, very fast. And you know this is clotting cascade. So this is like cascade of events are happening. Those factor 10 is getting activated, which is leading to another factor getting activated and just leading to clot formation. We saw this slide also, how hospital-acquired infections are issues. So I'm not going to talk about this. And how biofilms form, right? I just wanted to still give you like all the background because I know some people are new and not attended the last class. So bacteria adheres to a surface, it starts multiplying. Approximately how much time for bacteria to multiply?
17 minutes.
It depends on different strains of bacteria, but yes, around 20-30 minutes it's multiplying, and it's also forming EPS, extra polymeric substance, which is protecting bacteria. And the problem is now you have to give 1,000 times the dose of antibiotics because they are not penetrating those biofilms. And bacteria is learning how to not die with that antibiotic. They are getting resistant to those antibiotics, right? So all these challenges are right now there. So our vision, our human physiology is that our blood vessels release nitric oxide gas. That's one of the mechanisms for our blood or platelets not to get activated in the body. Okay, this nitric oxide gas production from our blood vessels. Also, when we are sick, our macrophages, one of the mechanisms to fight infection is it releases nitric oxide. So nitric oxide is helping fight infection also in the body, and it's fighting clotting also in the body, prevents platelet activation. So our body is using this, it's our physiology to fight clotting and infection. Existing technologies have antibiotics, silver, heparin, lot of these different technologies trying to fight clotting and infection. First of all, you can either fight clotting or infection, nothing is fighting both, right? Antibiotics cannot fight clotting, heparin cannot fight infection. Plus, antibiotics are learning to not die from the bacteria, they are developing resistance. Antibiotics cannot penetrate biofilms, so even with antibiotics, infection is still happening. With heparin, clotting is still happening to some extent. And there is 5% population which is allergic to heparin, and it can cause HIT or heparin-induced thrombocytopenia in those patients, right? So our vision is, why not we make surfaces Now, whatever it is, a catheter or a tubing or a vascular graft, whatever is getting implanted or going to see blood, can we make it nitric oxide releasing so that we can fight clotting and we can fight infection? And it's the same coating that can fight clotting and infection. Nitric oxide, so once it was considered a toxic gas, but in 1992 it was Molecule of the Year by Science. In 1998, these three gentlemen received a Nobel Prize for its signaling effect in cardiovascular systems. So it has been there. A lot of groups are trying to study it in different ways. Some are trying to understand the mechanism of it. Our group and some of my collaborators, I'm going to show some of them to you today. We are trying to design materials and make devices with those materials. This was just an interesting picture I saw online, that how our body, our blood vessels release nitric oxide gas, and that helps in our blood to properly circulate in the body. But as we grow older, the nitric oxide production keeps reducing, which can lead to thickening of arteries, inflammation, calcium buildup, and slowly as we grow older, the nitric oxide production is decreasing in our blood vessels, leading to more health and cardiovascular issues. Nitric oxide has a lot of different roles in our body. Okay, it can promote wound healing, it can help with angiogenesis, regulates blood pressure. The main two things that we are going to discuss today is its ability to inhibit platelet activation and its ability to kill bacteria. Okay, it's a neurotransmitter also. It helps in, I told you, angiogenesis, that's growth of new blood vessels, so you can have a faster healing of the wounds. So a lot of different applications, but we are going to focus on platelets and bacteria. Nitric oxide is produced in our endothelial cells, and there was this paper in 1998 by Dr. Mark Vaughan from Texas Tech Lubbock. He did a computational modeling and showed that this is approximately the amount of nitric oxide that our endothelial cells are releasing. Okay, so 0.5 to 4, 10 to the power minus 10 moles per centimeter square per minute, or we can call it 0.5 to 4 flux units. So we understand, see, the study was done 20, 30 years ago, and it was a computational modeling type study. So we understand this number might not be accurate, but it gives us a really good idea of what to target at least. When we are designing materials, we want them to release nitric oxide. We release. This helps us. And we are not just relying on this number, because we will test that release with bacteria and blood, and we will test that release against toxicity also, just to make sure that the levels of nitric oxide we are producing are effective in killing bacteria, are effective in preventing clotting, but they are not so high that we are killing healthy cells. Now, a question for you. You don't know anything about nitric oxide, and let's say you are starting the work in nitric oxide field in making catheters, let's say. What do you think are the challenges? I want to make a nitric oxide releasing catheter. What do you think are the challenges, can be the challenges with this field?
First would be toxicity. Too much of nitric oxide can turn into
So level of release, yes, okay. How not to have very high release?
So reactive nitrogen species in body causes a lot of inflammation and interferes with the body's immunity responses. So that could be another challenge.
Sure. So we have to really monitor the levels of release we have. Okay. What else?
How are we going to take some bacteria, whatever the component is, which is going to release to the surface of the...
Okay. So if we have some molecule that releases nitric oxide, how are we going to stick it to my surface, right? Whatever surface it is. Okay, that's a good one. What else?
So if our primary, whatever, because nitric oxide when exposed to the environment, we have to air water. So ultimately in our body, if we are implanting something which is releasing nitric oxide, when it comes in contact with water, it will turn into nitric. So we monitor that also. Whether oxide is actually working for us or nitric is actually working for us. That can be the thing.
Okay. Not couple of decades, we won't be able to do that, but yes, the longevity of release, like we don't know how much that is, but depending on the application, 1 week, 1 month, some number of days. Okay, what else? Forget nitric oxide. If you're designing any catheter, what do you think are the challenges? Sure, we want to make it anti-clotting, anti-infective, sure. But what can be other challenges? You know, you cannot say biostable in exam tomorrow. Okay, very good. How are the physical properties of the material going to get impacted, right? Because after you put nitric oxide donor in your polymer, are your strength is going to decrease by half, or right? That's not desirable, won't be accepted. What else? Actually, you are very right. So that's a good point. We did a study in that ECC model I showed you yesterday, where there is a tubing in the rabbit, and our coating was not good, and lot of nitric oxide was leaching in blood from the donor molecule that we were sticking in the polymer. It was not done properly, and that really dropped rabbit's blood pressure a lot, because we had like lot of bolus of nitric oxide release happening. So do you think storage stability, right? Because when you're making a catheter, it's not going to be used tomorrow. It might be used after 6 months. Also, let's say they are making the catheters in Jaipur and it's so hot there, it's coming on trucks. In 2 or 3 days, it's reaching different places in India in that hot environment. So it's not just storage stability in freezer or refrigerator, but decent stability during transportation also, right? Okay, what else? Sterilizability. We talked yesterday. If we sterilize it and it's either chemicals or heat are destroying my surface, now that's a problem. And really big one, expenses. If it's very expensive, it's not going to become a practical solution, right? So when I started working in this field, these were some of the challenges. How can we get a long-term NO release? Can we get stable and manufacturing ability of this material? Can I store it and sterilize it? Nitric oxide donors are heat sensitive. Okay, so when they are subjected to high temperatures, for example, extrusion is done at a very high temperature. It will just destroy the material. If I do 120 degrees Celsius sterilization, it will kill the material. And another thing we will talk later, nitric oxide can kill bacteria, it can prevent clotting, but one of the limitation is nitric oxide releasing surfaces do not prevent protein adsorption. Proteins come and stick to the surface, and now nitric oxide is fighting platelets and bacteria, but that layer of protein is getting deposited on the surface. Bacteria is also sticking on the surface. Nitric oxide is killing it, but nitric oxide is not anti-fouling. It's not preventing stuff to stick on the surface. Okay, so then there are two types of, when we talk about nitric oxide releasing materials, there are two different ways to do it. One way is called nitric oxide releasing material, and I'll have a lot of examples I'll show you. And one is called nitric oxide generating materials. Okay, so the difference is, in nitric oxide releasing materials, we have nitric oxide donor molecules. We synthesize some molecule that has nitric oxide bound to it. We can put those molecules inside the polymers, and I'll show you some methods we do it. And now slowly that nitric oxide gets released. Okay, we will talk what is a trigger. That's one method of doing it, and that's called nitric oxide releasing materials. Now, nitric oxide generating materials, or no-gen, is when we put a catalyst in the coating, which can be copper or selenium, for example, and our blood has these packets or RSNOs or S-nitrosothiols that carry nitric oxide. When this packet of nitric oxide carrying molecule comes in contact with this catalyst, it releases nitric oxide. So we are not relying on the coating that has nitric oxide donor in it. We are relying on the nitric oxide packets present in blood, and our catalyst is converting that packet to nitric oxide release. Okay, so nitric oxide releasing materials, nitric oxide generating materials. Okay, I think I have another slide where I have, in my 3-hour sleep, I thought I deleted this slide because I have more slides later, but I did not. So let's see how many other mistakes I have made. But this is not a mistake. This is one of the papers that talks about nitric oxide generating materials. There are more papers later, and the idea is same, but I wanted to put all the papers together. So, okay. Now let's talk actually about nitric oxide releasing materials first. Okay. Like you said, that there is a molecule that releases nitric oxide, right? And that's correct. There are different types of nitric oxide donors. Donor molecules that have nitric oxide bound to them, and they can release nitric oxide. So one such donor molecule is called diazonium dilates. Okay, or in short, we will call it DBHD. Okay. In our lab, we have nitric oxide analyzers. Basically, what this instrument does is, it can very accurately and selectively measure nitric oxide release from the surfaces. So, for example, I make my catheter or film or wound dressing or whatever, put it at 37 degrees Celsius in a buffer. I can accurately measure how much nitric oxide this surface is releasing. Okay, and the number I can get is that 0.5 to 4, 10 to the power minus 10 moles per centimeter square per minute. We can get that same type of number with that instrument. So we know, am I releasing 1 flux or 2 flux or whatever flux I can release. So I can very accurately measure it. Now, diazonium dilates. So don't worry about this yellow or orange, whatever, don't worry about that. Basically, when I started working in this field 15 years ago, what was happening was, we have nitric oxide donor diazonium dilate. Let's say we put it in the polymer, we will talk about it. And in presence of buffer and 37 degrees Celsius temperature, it will break and release nitric oxide. Okay, but it also makes diamine species. So there is nitric oxide released and there is diamine byproduct formed. Now what happens is when this diamine byproduct is formed, it makes the pH go up. So if I have a coating, let's say I have a polymer, polyurethane, I dissolve it in THF, a tetrahydrofuran or a solvent. Now I have a solution of polymer. Now I put desired amount of diazonium dilate in that solution, dissolve it, and now make it in a form of a film or a catheter. Now I know I have put 1 milligram of diazonium dilate in the polymer. Okay. But when I put this film in my nitric oxide analyzer with that 7.4 pH, 37 degrees Celsius, nitric oxide will release, but this byproduct of diamine species will form, and I only get this much nitric oxide release. So within 20 hours, so when I started working in this field, within 24 hours, the release will die. Okay, y-axis is nitric oxide release amount, and the x-axis is time. So, but I put diazonium dilate worth of, let's say, 5 days of release, and my release is dying in 24 hours. So this was the issue when I started working in the field. Okay. So one thing is, whatever I'm showing today, it's all teamwork. You know, my mentors help, my collaborators help, my students help, my postdocs help. So it's nothing that my work only, it's like we all work together. Somebody is a chemist, say they gave a really good idea. Somebody is a microbiologist, they gave a great idea. So that's how science moves forward. Okay, so we talked about this. Now, during my PhD, I published, so this is important for all of you. Lot of time we think, oh, my PhD is in this area, I'm never going to use this again in my life. I don't need to pay attention. You never know. So in my PhD, I worked with this professor in pharmacy, Dr. Jayant Panyam, on some extra projects other than my PhD projects. He was a big drug delivery guy with PLGA. I didn't know what is PLGA at that time. So I worked with him on PLGA drug delivery type applications. So I remembered that we talked how PLGA can break to form lactic acid and glycolic acid, and PLGA is FDA approved. It's not toxic, right? You can have PLGA that degrades very fast. You can have PLGA that degrades very slow. And it's very soluble in lot of solvents. So, and this was like 3-4 years ago I did this work, right? So then we discussed and thought, why don't we use and put PLGA in our coating? Okay, so now I have a catheter material. I put my nitric oxide donor in it. What if I put little bit PLGA in it? See, I know for a catheter, I don't want a biodegradable material, right? But if let's say 5% of my coating is PLGA, 95% is polyurethane for strength and whatever, right? What will happen is, what was our assumption was, our hypothesis, that when this reaction will happen, when diazonium dilate, my nitric oxide donor will break and form diapine. At the same time, PLGA will also break and make acids. And don't worry, I know you're thinking, oh my goodness, your acid in the blood will change. We are talking at like very minute amounts, right? Only acting inside the coating. So at the same time you're making diamine, but at the same time you're forming these acids, and these acids and diamines can neutralize each other, and the coating pH can remain neutral or slightly acidic. So this system can keep releasing nitric oxide. Okay. And see, I'm showing you like 2 years of work, but thinking is one thing, making it happen is another thing, and then you are not finding the right PLGA is another thing. So there is a lot of optimization. Okay, so what we did is we designed our coating, and And let's say we use polyurethane. We tried a lot of different polymers, PVC, silicon, rubber, polyurethane, but let's say polyurethane. So we made a polyurethane film. We added polyurethane, disanium dilate, PLGA in it. But then we put a top coat of only polyurethane. Any reason why we did a top coat of that polyurethane? And see, I ask these questions to force you to think. I'm not expecting you guys know this, but I'm just trying to forcing you to think a little bit. Active layer is inside, and then outside top coat is just the polymer. So my top coat is not PLGA. My top coat is only polyurethane. My top coat is not degrading. It's not PLGA. PLGA is inside. The top outside coat is just polyurethane. Okay. And what else? Yes, that's fine. What else? That's good. And also remember, let's say you have a catheter. If my outside PLGA is there and it starts degrading, I'm making tiny structures on the surface, right? It's protected now inside. Even if it's little bit breaking, 5%, outside is not seeing anything of that little bit holes in the surface, or not holes or whatever, right? Another reason is we want to reduce leaching of my anode donor. See, I want my anode donor to keep releasing nitric oxide in presence of like an acidic environment, 37 degrees, moisture, but I don't want my whole anode donor molecule to come out. See, if a lot of molecule comes out, it's going to be toxic, it's not going to last long, it will release a lot at the same time. So it's protecting it inside and not letting it leach out. Also, you are smoothing the surface from outside, little bit we talked, and you are neutralizing the surface charge because of disanium dilate. Okay. So when we were designing these surfaces, we thought a lot of things. So if you were designing this surface, what all would you think? This is your first PhD project. You are designing a coating with PLGA, polymer. What all will you think about when you're doing this? A lot of time in manager will meet with you and give you like a big idea. You can break, and in the acidic environment you can have disanium dilate. Okay, this is your first PhD project. Thinking and designing a project. What all do you think about it? What thought will come to you? Like, okay, now you are going back to your lab and now you have to start doing something. What all would you think? Okay, and remember, we are not releasing DBHD. That's an important thing to remember. I know you didn't mean that, but I'm just trying to again and again say, we never want anode donor to come out. We just want the gaseous nitric oxide to come out. And it's very easy for nitric oxide to just get released and come out. Okay. Anybody else? So NO it's a small gaseous molecule. It can penetrate through all these polymers. Okay. I also forgot to mention nitric oxide has another benefit. It can penetrate biofilms. Because it's a small gaseous molecule, antibiotics have difficulty penetrating biofilms. Nitric oxide can penetrate biofilms. Okay, sorry, that was off topic. What else? This is what we did, right? This is why it took us so much time. Thickness of the coating. But also remember, in the real life catheter example, we cannot play too much with the thickness, right? Because we need to have certain inner diameter, we need to have certain outer diameter, and we have very little room to play with the thickness. But you are right. Can we make active layer very thick, top layer very thin? Compared to each other, how much we can play. So let's say, I'm just saying example. Let's say 800 micron is my total thickness allowed. Should it be 400, 400, 200, 600, 600, 200? So we played with that a lot. Okay. What else? Very good. So we want to think, what should be our base polymer be? Like, should we use PVC? Should we use polyurethane? if polyurethane, what type of polyurethane? So different polymers have different water uptake. And in this type of application, water uptake is very important. Why? If it's hydrophobic, that's an issue because water is not penetrating inside. Until water will go inside, this coating won't start breaking or releasing, not breaking. So we want it to be little hydrophilic. But if it's too hydrophilic, that's a problem. All the NO donor might leach out of the coating, and that's a problem. So we want a balance of how much water uptake is there or how little water uptake is there. So we have to decide the polymer. What else? It's all there. Huh? Sure, but what else? It's there. We are not even worried about testing. I'm just thinking you're going to lab and you want to start making your coatings. What all are you thinking? Smoothness, we know if we'll do the top coat, we have smooth surface. Do you think you have to think about how much Dysanium dilate to add?
Yes, quantity of each of
The quantity of PLGA and quantity of Dysanium dilate
and the polymer.
and polymer. I mean, polymer will be maximum
Yes.
because that's giving the structure and strength to the material. Other two things are, see, PLGA has to be very less because we don't want it to start dissolving and your catheter is dissolving now, right? Dysanium dilates, if it's like a powder that's nitric oxide releasing. If you add too much Dysanium dilate in a catheter material, it might start becoming brittle. So now we had to play with what's the ratio of all these things. Okay. Then another thing was which type of PLGA? Do we want a very fast degrading PLGA or do we want a very slow degrading PLGA? Okay, so these were some of our drawing board things to do. Okay, and see, it sounds okay, but if you start doing it and start making coatings and then start testing, you want to test NO release also, you want to look at the strength also, you want to look at the leaching also. It can become a lot to then narrow it down that what's the right material. Okay.
So, do you use any kind of binding material also to get all these DHT, PLGA to stick to a base layer?
No, so it's just suspended in that coating for this work.
So, if we are, let's say, if we are designing a catheter that would go in the market, so will there a binding material required?
So yes and no. We can just suspend it. We can covalently bind it to the polymer chains. We have tried all these different things. I'll show you. So there is, yes, if you don't bind it, you want to be sure it's not leaching out. If you covalently bind it, now it's a little bit safe because it's not going to just leach out. It's bound tightly, right?
I suspect that there is polymer, like the coating of all of these materials.
You can do that also, or you can make a solution and mix everything up and then cast a film, or you can do layer by layer coating. So for example, this is off topic. When we make catheters, what we do is we take a solution, whatever that solution is with NO donor or whatever, and we take a metal mandrel and we dip coat it. Let's say one dip coat every minute. So I dip coat it, wait 1 minute, dip coat it again, wait 1 minute, do 30 dip coats, 40 dip coats, 50 dip coats, let everything dry. Basically, dip coating has put lot of layers of that polymer on that mandrel. Remove the mandrel, you have a tubing or a catheter ready. You can have first few dip coats of your control material of your base polymer. Then you can do some dip coats of your active coating Then you can again do some dip coats of your control material. Then in that mandrel, your inside first coat was just base material, then dip coats of your active material, then dip coat of your base material. So when you remove the mandrel, inside and outside of the catheter is base polymer, sandwiching your active material in the middle. Right?
So, like, so, picture we can see it's just fingered in the same way. There are multiple signs that prolong the
We have tried a lot of things. So multiple sandwiches you mean is one coat, top coat, another coat, right? See, that's a good idea, but thickness is a limitation. Now, you can add another coating also if the polymers are very different from each other.
It's the same polymer. Uniformity could be there in the polymer. The active coat will also remain the same. It's just multiple emitters.
Oh yeah,
And
yeah,
thinner than this.
yeah,
Not this thin.
yeah. Of course, you can play with all that.
So like micro of that polymer, and we can have like 150 or 200, and we can do things.
So you are saying layer by layer. You have NO release layer, control layer, NO release layer, control layer. Yeah, yeah, yeah. See,
But so if we're doing this and NO layer and then when the NO is released,
It can release its gas.
it can come out. Yes, it can come out. And see,
no option.
you are on the right path. This is how people in my group think. Like, this is the way we think and design new materials.
So we always consider the limitation restriction first,
Yes.
and then think around it.
Yeah, and then thinking out of the box, because so many people are doing so many things, right? Some are our collaborators, some are our other scientists who we don't work with. Everybody's trying all this, and the goal is same: how to make a stable material, control release, longer term release. Okay. Then, forget borate. I don't want to tell you the borate story. That will take some time. So forget borate. Forget it. Now, this is an example showing when we did just a polymer with dizanium dilate with a top coat of polymer, this green is what happens with the NO release. In 3 days, NO release is dead. Because my coating is becoming so basic that release is stopping. Okay? But then when I add PLGA in this coating, the red one, I can get release for 14 days. So the idea we had, hypothesis was right, that you can add little bit PLGA, it's keeping the environment little bit acidic, little bit neutral, and you keep releasing nitric oxide. Okay. Then, yes.
So why is this borate and control missing in 7?
Oh, it didn't last.
Okay, it's exhausted.
It exhausted. Yes, yes.
And so control here is?
Control is your polymer, same polymer, has dizanium dilate in it, DBHT, and a top coat of polymer. Your active is everything same, but with PLGA.
PLGA is missing.
Yes, yes. Control and borate have no PLGA. Don't even worry about borate. But control has no PLGA, and your PLGA is the only additional thing you have.
One more question. Like few slides back, you showed that the blue and yellow lines dotted graph.
Yeah.
Some borate, tetraphenyl
Yeah.
borate additive, but it was toxic. It was extending the release, it was toxic. How
Yeah.
do we deal with that?
That's why we cannot use it.
We
That's why I
cannot
didn't even want to go in that story. So that was being used when I started working in the field a little bit,
Okay, just to explain it.
but I didn't want to go in that direction. So I just, yeah. Okay, then what we did is, we got different types of PLGA. Okay, a very fast degrading PLGA and very slow degrading PLGA. 1 to 2 week degradation and 1 to 2 month degradation. And when we did NO release, see, this is interesting now. You have to pay a little bit attention. Black is 1 to 2 week degradation PLGA. Okay, black. So that means it's degrading very fast. That means it's creating very acidic environment in the coating. That's why the release is so fast on day 1. But it's dying by day 10 because you released all your diazonium dilates very fast, NO release very fast, and it died quickly. But when we did 1 to 2 month PLGA, we had a more consistent release for a little bit longer period of time. I don't remember now. Maybe we tried 1 to 2 year PLGA also, and maybe that was too slow and we couldn't see the effect of it.
One question.
Yes.
In the black one, since it's usually acid very fast, what will cause, since the polymer is a kind of metal only, some, not a metal, but would it cause a acidic corrosion or something? It's too fast?
Not acidic corrosion, but can be toxic. Like the levels of nitric oxide release are very high, more than 10 times what that computational modeling number showed. It might have been. See, the thing is how science moves. I published this paper 10, 12 years ago when I was working with my PhD, not PhD mentor, Dr. Bartlett. We were not doing toxicity studies at that time. We didn't have the facility to do toxicity studies, so we never tested this material for toxicity. But it could have been toxic, you know.
So what's the difference? 1 to week PLGA, 1 to month, or 1 to year?
So PLGA is polylactic glycolic acid. The ratio of lactic to glycolic acid can make it a very fast degrading or very slow degrading PLGA.
So on the third day in that graph, the PLGA, second PLGA, 1 to 2 months, it rises. The drug release. So it is due to that the degradation is happening, or
So first of all, this much little bit up and down, it's okay, you know, but next slide will show you what's happening. Okay, let's pay attention to this slide. This was very interesting also. So there is this dye called bromothymol blue. What this dye does is, if you add this dye to a very acidic solution, the solution will look yellow. If you add it to a basic solution, your solution will turn blue. Okay. So what we did, okay, we think this is what's happening, right? That PLGA is breaking, acidic environment is creating, diazonium dilate is breaking, basic environment is forming. This is what we think. We wanted to show it more qualitatively. Okay. So now what we did, pay a little attention again. We added little bit of bromothymol blue dye in our coatings. Okay. Let's see control, which has no PLGA. You start with a bluish little yellow tint coating. It releases a little bit nitric oxide for a day or two, makes it more basic. Your blue color becomes darker in the coating. When we had 1 to 2 week PLGA, your coating is yellow because your PLGA is breaking so fast that it's making very acidic environment inside the coating. But that's releasing nitric oxide, making some diamine species, making a bluish, pushing it to bluish color because it's fighting to make it basic environment. That's why it's landing green color, because it's somewhere in between yellow and blue. But by the time it's day 7, all the nitric oxide is gone. And now PLGA won the battle. PLGA is making it acidic. There is no nitric oxide donor left to make it basic anymore. That's why it turned yellow, that it's done. And that's what it matches here also, that by the time it's day 7, day 10, your NO release is dead. But when we had this 1 to 2 month PLGA, the coating remained greenish for a very long period of time, because there was a very nice balance between NO release and PLGA breaking. And see, a really good application of this is if you have a catheter in a hospital setting and you tell the doctor if this catheter turns yellow color, cap or something, that means NO release has died. That means all the NO donor is gone and you just have a yellow color now. Okay. So this was the story of diazonium dilate molecule, nitric oxide molecule. Okay. But later we started working a lot with SNAP or S-nitroso N-acetyl penicillamine. This is another NO donor molecule. Okay, diazonium dilate is one type, which is little bit more complicated to synthesize. You need a high pressure system in the lab and everything. We have it, but this one is little bit more convenient to work with. Now, SNAP is also synthesized from NAP or N-acetyl penicillamine, which is actually FDA approved for heavy metal poisoning. So our idea is also, if we are using something that's FDA approved and synthesizing SNAP, maybe going and getting a FDA approval might be a little bit easy to go that route. Okay. Now, unlike diazonium dilates, we don't worry about acidic or basic environment here. Heat, light, metal ions, moisture, those are the triggers for nitric oxide release. Okay. But then, like heat is a trigger also, but that means it's heat sensitive also. Even diazonium dilate is heat sensitive also. And that's important, right? That's why at a body temperature, it starts breaking and releasing nitric oxide. So we want it to be little bit heat sensitive also, or break with heat or light or metal ions. Okay, so with this work, when we started working, we thought we again wanted to go to the same question, which polymer to use. So we tried lot of different polymers with different water uptake. Silicon rubber, carboxyl is a copolymer of polyurethane and silicon rubber. E2AS is a copolymer of polyurethane and silicon rubber. SP60D60 and SG80A are polyurethanes, just two different types of polyurethanes. Okay. Now, but remember one thing, the top three polymers have very low water uptake, 1%-ish. And the bottom two have very high water uptake, 6% and 60% water uptake. So what we did first study was, we made this solution of different polymers, put SNAP in it. SNAP is a NO donor. We don't need PLGA and all that now, so forget PLGA now. And we casted films. Put them in buffer for 24 hours. And what we saw with UV vis is that SG80A SP60D60, all the NO donor leached out within 24 hours. Very fast. In 4 to 8 hours, most of the NO donor was leached out. So those are not good candidates for us. I don't want my NO donor to leach out. But the other three were really good. They almost kept all the NO donor inside. Because the water uptake was so high, water was entering inside and bringing all the NO donor out. This is just an example showing how we can switch on the light and increase the NO. We can switch off the light and reduce the nitric oxide level. A good example of this can be, for example, wound healing. You know, you have a dressing. If you feel every day, oh, infection can happen, your nitric oxide levels are only 0.5 or 1, shine light for few minutes or 1 hour or so that you can have higher release of NO happening and you can take care of infection and then you can switch it off. Okay. Another idea, we have published a paper also, but I don't think so I have a slide on it. It's a little bit off. We have not reached there yet, but I will forget if I won't tell you. So we have also done make a nitric oxide releasing snap catheter that can be implanted, but have fiber optic cable that you can insert inside this catheter. Because fiber optic will release light, that light will trigger more nitric oxide release. So you can have that fiber optic thing going on for few minutes or few hours, or you can even have like a short fiber optic, because a lot of time ports that you are using can get infected. You can have a port connected with the fiber optic. After you take the blood out or infuse something, put this cap on with fiber optic so that it can release more nitric oxide and take care of infection if there was any infection, higher levels. With this snap method, we were able to prolong the release to 20 days. See, first when we started, we were at 1 or 2 days of nitric oxide release. Dizanium dilate helped us to reach 14 days of nitric oxide release. SNAP helped us to reach 20 days of nitric oxide release. Yes, yes. So a lot of time. So yes. So these materials, they can be light sensitive also. So a lot of time we just store them in dark places. But that's easy to take care of. You can have a packaging material that's dark, like opaque.
So no, I mean, I'm even so I insert a catheter.
But see, insert is, it's inside, right? So that light is not reaching inside. Right?
So when you were testing this light sensitivity with optical fibers, the problem that I see is that is only at a specific point. Or something.
So we can, right? You know, if you think the port might get infected, we can have a short fiber just there. If you think it's longer, we can just put a little longer fiber inside. You know, we have lock solution. So instead of a lock solution, you have this lock fiber optic cable, battery operated, that you can have. Or you can only have it for few minutes or few hours. Immediately after a procedure, have that cap for 1 hour, have the light on, and then after 1 hour you can switch off the light, you're fine. If any bacteria was introduced during the procedure, you took care of it.
So,
Yes.
so what method do you use to keep check on the time for which the light is being released by that optical?
So, you know, with the nitric oxide analyzer we can do all the in vitro study, right? That how much time, what intensity light we have on, what are the levels of nitric oxide release.
If the catheter have shelf life. So I mean, if it is releasing nitric oxide and suppose it's as you said the time is for 20 days and it is packed and it is kept like that. If I've not used it, then also the NO will deplete.
No. So 20 days is testing at 37 degrees in buffer. If it's actually, it's coming in 2 slides or 3 slides, I think. When we are storing it in refrigerator or freezer or on the shelf, it's not releasing, maybe it's releasing less than 5% nitric oxide because it's not sitting at 37 degrees in moisture all that time.
We have to take care of the conditions it is kept in so that it does not start releasing.
Yes. Of course, if you put that catheter in liquid, then nobody will use that catheter anyways. But if it's kept at room temperature or in fridge or in freezer, in refrigerator, freezer, it won't even release anything. At room temperature, dry conditions, it might release 5% of it. It's coming in 2-3 slides, that data. Okay, so Marcus was my first PhD student and he did some work where he tested a lot of these things from commercial side of things, you know, and I'll show you a little bit of this from Marcus's studies or other studies. But the point was, how are the surface properties, how are the mechanical strength, sterilization effects, NO release. And I have put the article, if you're really interested in a little bit of that, there is an article, you can read all that, but he did a lot of studies like that. Storage stability, so lot of those things. This is another good slide. Actually, this slide is 10 years of our work. I've tried to show it in this way. In 2009, when I started working in this field, we used to have 3 days of nitric oxide release. I showed you already, right, without PLGA. Then we published a paper with PLGA1 and we reached 14 days of nitric oxide release in 2012. In 2015, we had 28 days of nitric oxide release with some snap things. But then in 2018, we published a paper where we had a 4-month of NO release. So to give you an idea, if you show it to somebody, it looks like one graph, but this is a journey of 10 years to go from 3 days to 125 days of nitric oxide release. So this material was silicon rubber, I think, PDMS. So let me go back a little bit. So this graph is a little bit misleading in a way. This was all silicon rubber data. I have just shown you, this is all one material of 125 days. I'm just showing you that in our goal to achieve longer term NO release, different materials we tried, different combinations we tried. In 2009, we reached this goal. 2012, we reached this goal. Something was done with diazonium dilate, something was done with SNAP, something was done with PLGA added, but then we were able to achieve 125 days of release. Okay, so this is your question, Neha. We did a shelf life storage study. Actually, we even have a data of 6 months now. I just didn't put that data here, but we have 6 months data, that how in freezer and dark and room temperature and 37 degrees Celsius, how much NO we are losing. But it's all in dry conditions. So we lose less than 15% of NO. And actually with the newer materials we have, the 6 month data, we are losing only less than 10% of NO in storage while shelf life. We did sterilization study. Actually, this is Marcus's paper also. He has some of this data. That we tried different sterilization methods, autoclaving to UV to ethylene oxide to plasma. Autoclaving was bad. Everything else was fine. We were able to have more than 90% NO release capabilities.
So what exactly happened at 50 degrees Celsius in that graph?
In what happened at 37 degrees?
50 degrees Celsius.
50 degree. Oh, I think maybe we didn't show it because maybe it dropped more. So we didn't do. Also, I think our thought was also now, I think, we only did 1 month of 50 degree, thinking if we are shipping something and now it's stuck and sitting in a truck in Delhi for 1 week, then temperature in that truck might reach in 40 degrees or 45 degrees. So that's why we only did 1 month. It better not be sitting in a truck for 6 months. If the truck is delayed or something, that's why we only did for a month of that very high temperature sitting. Yeah.
No, but actually my question was, what happened with that?
What happened means after,
Like for 37 degrees Celsius, it is prolonged for 4 months. But in 1 month, the 50 degrees Celsius, it dropped so much. Why?
yeah, because temperature was high, right? We are losing more nitric oxide.
Oh.
And also, we are not going to store those catheters at 50 degrees. I mean, that will be ridiculous to store it in outside in sunlight.
Within a month at 50 or so petrol that we did, it was not.
Also, this study we did only to prove that if by chance a catheter is getting, see, catheter should be sitting in a room temperature on a shelf or let's say in refrigerator. But if you are shipping it, it better reach wherever you are sending it within a week or two. Worst case, it's a month. You might lose 15% of NO, but then you can keep it in the refrigerator and you will maintain that level now. Right? It should not just be sitting at 50 degrees for 3 months. We should not, and if that's the condition, then maybe this is not the best
So
technology.
suppose if it was 1 month,
Yeah.
it lost some amount of NO. So it will also reduce the shelf life of the catheter, as in that after 1 month, it can only be used for a certain period of time. And
So-
because they have already lost, suppose 50% of NO, and there's only 50% left, and
Yeah.
we can only use it for some.
True. Also, see, remember, if we market this type of catheter, we are not going to say 120 days. There'll always be some buffer. See, if a medicine expires in December, you might actually label September that, hey, medicine, that like everybody's keeping that extra buffer that you're not saying absolutely, oh, my catheter is 120 days. Maybe we will only market it 60 days. So you are already considering those things. And I think if we go forward in that direction, maybe FDA will ask those type of questions. That, okay, if you lose 15% of NO release, and I'm almost sure we already have that data also. Somebody must have done it. If you have lost 15%, now what's your NO release trend? Are you releasing less every day but still lasting 120 days? Or now you're lasting only 100 days and not going 120 days. So all those things will have to be discussed, right?
So, why do you think in all the four situations that we are keeping on the 15th or the 20th day, there is a sharp dip, and then throughout the duration, there is not a significant change drop in the material?
Okay, that's a very good question. So we found out that the SNAP NO releasing materials, they form crystals inside a polymer. Okay. But some of the SNAP is not crystallized, little bit. So what is not crystallized, that can come out fast. But what's crystallized now, that will release very slowly. When moisture will go inside, it will try to little bit dissolve the crystal and bring little bit NO out. So you saw that little sharp down because of that uncrystallized SNAP that's sitting inside the polymer, that came out faster. Mostly for any NO release polymers when we make, we see that with NO release also. Lot of times we see little bit higher NO release on day 1 or 2, and then it starts going little down because of that uncrystallized SNAP that's just sitting around there.
It's actually good that it's very crystallized.
Yes.
Preserving for longer.
So that's why we were very happy when we got 20 days of NO release. That's stabilizing SNAP inside the polymer. Otherwise, the NO donor can just run out, but it's giving it a more. So since you started that conversation, we also found out if we put up to 10 weight% SNAP, or sorry, up to 5 weight% SNAP, it does not crystallize. But after that, some amount, 4 or 5 or 6 weight% SNAP by weight, so 95% polymer or 90% polymer, that's the quantity you need for the crystals to start forming. Okay.
So we are making sure that we use quantity in that range.
Yes, but we also showed if you add 15 weight% snap or 20 weight% snap, it will happen,
But less of it.
but your material becomes less strong and it starts becoming a little bit brittle. You are compromising on the strength of the material by adding too much NO donor. Otherwise, you can always say, hey, let's add 50% snap. That's great. We can have more NO release, but no, the strength of your material will go down really tremendously. Okay, so next interesting thing is, oh my God, today I think I need to drink more water. Thirsty. So next interesting thing was, so Professor Brisbois is working, I'm working with her for last 10, 12 years. She's a collaborator. I told you these NO donor molecules are very, if I sit here, it's okay, right? I can sit? Okay, great. Oh my goodness. Oh, okay. So, NO donor molecules are very heat sensitive. But when we are, I mean, I can stand for another.
So, sir, I mean, you can sit here.
Oh, okay. This is better. Okay. Great. Okay. So, NO donor molecules are very heat sensitive. But how do we extrude now? To make catheters, tubings, there is extruder, which is like pellets of polymers are dissolving. You are putting NO donor in it at high temperature and it's forming into tubings or whatever. But we saw autoclave. Autoclave basically killed all the NO release because of high temperature. So this paper was very good in Acta Biomaterialia. What we showed here was that if we swell polymer tubings, so what we do is we have a solvent with NO donor dissolve in it. We take existing catheters or existing tubings, swell it in that solution for let's say 24 hours. Idea is that solvent will take the NO donor molecule into the walls of the catheters, take the catheter or tubing out, dry it overnight or 2 days or whatever with vacuum to make sure you get all the solvent out. And now all the solvent is gone, all the NO donor is inside the wall of the tubing, and now you have a NO releasing material ready without extruding, without making a film or whatever, and it's very uniformly distributed inside the wall of the tubing, and you have a NO releasing surface ready. And
I
you,
had this doubt that water, instead of we can just make it.
yes. So now you can pick an existing tubing, existing catheter, and make it NO releasing. This paper, like, I mean, you can look at it. I don't even remember. Oh, we did even rabbit study with it. I just wanted to tell you the idea. We did a lot of studies to make sure NO release was good. The surface in the SEM looks fine. It's not that surface is becoming rougher now. We tested it in the animal model. So we did all that with this. See, I have a lot of slides like this now. Okay. One slide, one paper, basically. So I'm just giving you a basic idea. If you are really interested about the idea, you can go to that paper. Okay. I ran out of time when I was making the slides because I wanted to make like this. I have a lot of slides like this, but the paper information is there, the manuscript, so you can find it. So other idea was, we always have this reservoir of nitric oxide donor in the polymer. If it dies, my nitric oxide release will die, right? So we thought, why not we use dual lumen catheters? So there is a wall in between a catheter. It has two sides. Fill one of the side of the catheter with nitric oxide donor with some hydrogel, like PEG or whatever, and seal it. Now the idea is NO release that's happening from that lumen is protecting, NO release is going in all directions. It's protecting the other lumen that you are using to take the blood out or infuse something. Another beauty of this is every few days, let's say NO release from this lumen is 14 days. After 14 days, you can put a needle in this lumen, take this hydrogel out and refill it. Now you don't not worried about, oh, my NO donor will run out. Okay, so this was another paper. This is just an idea, like we wanted to just show it in a, instead of showing it in lumen, we made catheters, we filled NO donor in it, and we showed that how when we just fill SNAP powder in that lumen, we get a very low NO release. Okay, but if we fill PEG, which is a hydrogel with NO donor, we get a really good NO release because it's a moisture environment and hydrogel is helping NO to break and release. And here we are also seeing nitric oxide donor SNAP is green in color, that synthesized molecule. Over 14 days, how that green color is going away. So by the time it's almost 14 days, all my NO donor is gone, and I can refill the new one. Actually, I'm so sorry, I've said this was NO release. This was not, I just saw. This is water uptake. So of course, if you have more water uptake, you can release more nitric oxide. So with only SNAP powder, you have water uptake is very low in that lumen. But if you have a PEG in there, you are helping with more water uptake, which can help in more nitric oxide release. Okay. We already talked about this. Antimicrobial testing, bioreactor, you can homogenize a sample and then you can plate it. But this is just one study with one of these materials. This is not manuscript under preparation, it must be already published. When we make nitric oxide releasing materials and do a 7-day bioreactor study and do live staining, live dead staining, lot of green bacteria, live bacteria, and mostly dead bacteria with NO release surface. Okay, now we have next chapter of this thing. You know, let me do one thing. This is like a whole story. Let me skip some slides and talk about some different things, because this I want to do it in continuation after lunch. Okay, so let me actually go to, and I'll tell you guys which slide number. So we did this work in around, oh, sorry, 86, yeah. We did this work in around 2013, 2014. Acetobacter baumannii is a big threat, and it's a very drug resistant bacteria for wound infections, multi-drug resistant bacteria. So what we did, the study was, we used Dizanium dilate material with PLGA. We made a wound dressing with that polyurethane, PLGA, Dizanium dilate. And what we did, we took a mouse model, we gave a little burn wound at some 50 degrees Celsius water or something, and then we infected that wound of the mouse with Acetobacter baumannii. And then for 24 hours, we applied our wound dressing, and for 24 hours, we applied a control wound dressing. And then after 24 hours, we homogenized that tissue and that dressing, and we looked at the infection. So we found 4 log reduction. So see, I hope you know, you remember we talked about it. 1 log reduction is 90%, 2 log reduction is 99%, So 4 log reduction is 99.99% bacterial infection was reduced. Another, and I cannot tell you the details a little bit here, filing for patent and all that. We made some wound dressings that are loaded with drugs, whatever drug we want, NO release or clotting agents or whatever, but the dressing is completely biodegradable. Because a lot of time what happens when somebody has a burn wound or whatever very bad wounds, if you are coming and changing dressing every day, whatever cells had started growing, you are removing it again to put a new dressing. So the idea is you don't need to remove anything, you just put a new dressing every other day or every day, and it's releasing the drug, but the dressing is also dissolving while releasing everything. Another property of NO is GSNO is another type of NO donor. So don't worry about it. SNAP, GSNO, similar family little bit. We also, NO helps in cell proliferation. So if there is a wound, so here we did, maybe you cannot see it very clearly. We had cells, we put a cut so that there is a place where there are no cells now, we removed it. And with the dressing, we showed how the cells grow closer to each other faster. So they are helping with the growth of these cells and faster healing of wounds. Last picture.
So this one.
Oh, this one. Yeah, okay.
Control, you see that there's this rectangular red box and then yellow inside linings are there. Is that place where the wound was made?
Yes, yes, yes.
There are no cells visible there.
Yeah, you know, this picture is not great, but yeah, there were cells. We removed the cells. So there is also now, instead of doing scratch, so you call it scratch test. Okay. Now you also have these, I don't know what to call it, these inserts that you can have the insert. So when cells are grown, they are not grown in that insert area. You remove the insert and now you see how fast you are healing that area, how it's coming closer to each other. You know?
Later, after 24 hours, we what's there?
Yes.
So there are cell growth over there.
Yes. Okay, let me show you something else because I do not want to start that chapter. Okay. Okay, let's talk about this a little bit. Katie did her master's with me. So she worked on endotracheal tubes. So we bought endotracheal tubes and she made them NO releasing, and she did a lot of studies. You can go to this article and read more, but we did bacteria studies, we did leaching of SNAP, we did tensile testing of the because we don't want the endotracheal tubes to become too brittle and less strong. So we did a lot of those studies to show we can make endotracheal tubes more antimicrobial. See, these were the slides I was making and I was half asleep. So there is no good order to this. And there doesn't need to be any order to this, honestly. They are all independent papers. But since our break is coming soon, I don't want to start a chapter that will not be. Let me show you now. Okay, this is a good one. So slide number 97. Okay. So Laurie did her PhD with me and graduated, I think, 2 years ago. So this study was, I really like this study because a lot of times, most papers we have and others have in the field, we always infect the medical device with 10 to the power 7 CFU bacteria per ml, approximately. You know, that's a very high load of bacteria. And lot of time we show, oh, my nitric oxide or whatever materials people are testing, we have 1 log reduction, 2 log reduction. So we thought, why don't we try different bacteria infection rates? You know, 10 to the power 3, 10 to the power 5, 10 to the power 7. And it was very interesting. We basically showed, read this paper if you're interested, we basically showed that if your infection is 10 to the power 3 CFU per ml, nitric oxide can take care of the whole infection. But when the infection is 10 to the power 7, it's so overwhelmed with bacteria, it's fighting, but it can only kill 90% or 95% or 99% because there is so much bacteria and there is not enough nitric oxide to take care of it. But in real life, most of the time when somebody gets infected, they are not going to get infected with that big bolus of bacteria. Mostly patients will get infected with 100,000, 10,000 bacteria at a time. And if we have this nitric oxide releasing devices, we are taking care of that infection very fast. So I like that paper because we wanted to see, yes, nitric oxide is not killing everything, but is it because it's overwhelmed, or lower number of bacteria will be fine and we can fight it? So that was that work. Okay, I guess we can have a break now and then we can finish it. But please remind me, we should talk a little bit about tomorrow's test later in the class today. So, okay guys, thank you so much.